Hello, boys and girls, and welcome to Mendelian Genetics. Today we are going to be talking about Gregor Mendel, the history of genetics, one trait being controlled by one gene. That is Mendelian genetics. And this is what we're going to go over. Now, if you look right here, this man that you guys see right here and right here, he is Gregor Mendel. He is what we call the father of genetics. Now, you'll see a couple of key vocabulary terms. If you want to look at it a little bit longer, these notes, PowerPoint notes, will be up on Google Classroom for you to look at. But here are some of the some of the vocabulary terms that we will be using very in depth today. To go over on this one, you see that we have P generation. This is the parent generation. It is used for true breeding, meaning that all of them are going to be self-fertilizing, at least when we talk about Mendel and his pea plants. F1, first filial. Filial means child. So these are the offspring of the parental or the P generation. And F2, second filial, this is the offspring from the F1 cross. So these are the grandchildren of the P generation. Now, Gregor Mendel, he was an Austrian monk. So he was a monk. He was from Austria in the 1800s, and he used only his mind to develop these laws and principles of genetics. And how he did so, the story goes, was that he was looking at pea plants. And he looked at these pea plants, saw that they were self-fertilizing, meaning they bred they did not breed with another plant. They fertilized themselves. And if we were to look at the male and female parts of a plant, you guys would be able to see how that would work out. Now, what you guys can see here is he did this by observing the following characteristics of pea color, whether they were green and yellow, pea shape, whether they were round or wrinkled, flower color, whether they were purple or white, the plant height, whether it was tall or short, the pod shape, because peas come in pods, and the pod color. Pod shape meaning round and wrinkled, and pod color being green and yellow again. So those are the 7 characteristics that he studied. And he made the following observations. Now, right here, what you guys see is with a round or wrinkled pea. Now, the parent generation, we know for a fact, is homozygous dominant because no matter what happened whenever he saw those peas breed, they were always round or they were always wrinkled. So he crossbred them by removing the male parts of one plant and using it to fertilize the female parts of another plant. And he saw that all of the children came out round. So all of the offspring became round, which was weird because you would think that it would be a mixture between this round wrinkled pea. Like, for example, with plant height, it was either they were tall or they were short. You would expect a middle height, but that's not what happened. He saw that they were all tall, and in this case, all round. And then when he allowed this one to self-breed, meaning it fertilized itself, he saw that 3 of them came out to be round, but 1 of them came out to be wrinkled. So we saw a 3-to-1 ratio. Now, how he did this, here you guys see that the stamens, the part that has this yellow bit right here, hold on, right here, this is the stamen. And this part right here, this is the female part or the carpel. So we have our male and female parts. Now, he used that to cross pollinate. So here we have our white and here we have our purple. And he used it to crossbreed or to hybridize the children, where we got all of the round pods and all purple flowers. And that was the first generation offspring. Now, pea plants are great for genetics because they have distinct heritable features with differing variant traits. Um, and that Mendel had a strict control of which plants bred with what, because they could either self-fertilize or they could cross by him removing the male parts and using it to fertilize a different female. In nature, pea plants typically self-fertilize, so fertilizing their ovum with their own sperm, but Mendel can move this by cross-pollination, by physically changing it. In a typical breeding experiment, he would hybridize two contrasting true breeding pea varieties. So he would get the F1 generation. And it was through his quantitative analysis, so his data analysis of the F2 plants, that revealed two laws that are principles of heredity: the law of segregation and the law of independent assortment. So the law of segregation says that when we form gametes, sperm, egg, etc., we form them through meiosis and we receive one allele or the other. They separate and you get only one or the other. You don't get both. The law of independent assortment states that two or more alleles will separate independently of each other when gametes are formed. So a allele for being tall will separate individually from an allele for being short. Like they separate independently of each other. It does not say that if I have this allele must come with me. So if I have this tall allele, this other allele will come with. They will separate independently of each other. So by the law of segregation, the two alleles for characteristics are packaged onto separate gametes, meaning that one's going to be on one chromosome and the other is going to be on the other. So if the blending model was correct, then we would have, with purple and white flowers, we would have pale purple flowers, or if it was correct with tall and short pea plants, we would have medium pea plants, but that's not what happened. Instead, we got all purple flowers just as equally purple as the original parent, or all tall plants, just as tall as their parent. So this is a Punnett square, and a Punnett square was first made by a man named, of course, Punnett. It was made in the 1900s, so years after Mendel, and he used this as a tool for calculating genetic probabilities. So what we'll do is we'll do this with the purple and white flowers. Purple was dominant, so we are going to use a big P for purple and a little p for white. I do not have a stylus yet, sorry. So, remembering with the law of independent assortment and the law of segregation, it says each of these are going to separate individually of each other. So we're going to start with this parent one, where mom or dad, your choice, one parent was homozygous dominant for purple, and the other one was homozygous recessive for white. Now, this first one, it's going to go down and across, down and across, down and across, down and across. Which, to show you with some different colors how it works, down and across, down and across, down and across, and our last one, That one's going across, and this one's going down. So they all go, so this one's going to go down, it's going to go down, 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 across, 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 across. And the reason it does that is because each of these are going to be one sperm and one egg, and they are going to fertilize to make one child. So, using a several step process, we can look at many different genetics. Here, we have the process is we determine the alleles of each parent. It's given as big T for a tall plant, little t for a short plant, and we're going to separate them amongst the Punnett square. So one above each row, one above each column. And then we're going to write the letter for each allele across or down each row and column. So the big T is going to go down the column into each square, and the little t is going to go across the row into each square. And the resulting mix is a genotype for a ratio of offspring. So here, every single one of these children or potential children are all heterozygous. It's a 1, 2, 3, 4, 4 out of 4, or 100% chance of this child being heterozygous for tall. And they carry that little allele. So we're going to take this a step further and cross the F1 offspring. And so we separate the alleles, a big T on this first column, a little t on this second column, a big T on this first row, a second T on this other row. And since each parent can contribute a big T and a little t, our Punnett square will look like this. We will have big T for the first one, little t for this one down here, and these two will be heterozygous. Now, if we were to look and count this out, we see that we get a 3 to 1 ratio of being tall to short. Need to get a stylus. So 3 to 1 ratio being tall to short, and that is called the phenotypic ratio. What the phenotypes look like. Now, the genotypic ratio, what the genes are, is we have one big T, two big T, and one little t. So it's a 1 to 2 to 1 ratio, and that is our genotypic ratio. Remember, genotypes show the alleles, phenotypes show the physical characteristics. So once more, we have a 3 to 1 ratio of the phenotypes, 3 tall to 1 short, and a 1 to 2 to 1 ratio of the genotypes. So 1 big T, 2 big T, and 1 little t. So when we look at Mendel's recordings, we see that there was 705 purple flowers in the F2 generation to 224 white flowers in the F2 generation, which gives us that 3 to 1 ratio. One more picture. Here we go. Our big purple flowers to the white flowers, giving us all purple flowers, which then gives us 3 purple flowers to 1 white flower. Here, the law of segregation states that the two alleles for each character segregate during gamete production. So if we were to do this with a coin, it would be a heads would be a big P and tails would be a little p. So if we cross two of them, so we would get a double-sided heads coin. 2 regular coins and one coin that is 2 tails. The law of independent assortment says that each allele will segregate independently of each other. So this is where we get something that looks like this, where we get the big G, big Y, where they can segregate this way, and you get a big G, big Y, and a little g, little y, little g, little y. Or they can segregate like this, where they have a big G, big Y, or big G, little y, and then get a big G, little y, a big G, little y, a little g, big Y, and a little g, big Y. And because this is 2 different chromosomes, this would be a 2 to the n ratio, which means we have 4 different possibilities of this of different segregation of independent assortment. Now, a test cross is where we breed a unknown genotype to a known phenotype to see what we get, like whether that parent must have been heterozygous or homozygous. And we do this with the dominant phenotype to a known genotype that is recessive. So we could see what that genotype could be. Because the recessive we know is always going to be recessive. A white flower always has to be little p when it comes to pea plants. So we can take this purple flower and breed it to the white flower and then look at the children. If all the children are purple, then we know that parent had to be big P, because that makes all the children heterozygous. Where if 2 of the children turn out white and 2 of the children turn out purple, that means that parent must have been heterozygous, because this big P is going to go here and a little p is going to go here. Same over here. And then we get a little p here, little p here, a little p here, little p here. And we see that 50% are white and 50% are purple. So a test cross designed to reveal the genotype of an organism. The rule of multiplication is a rule that states that the probability that 2 events will occur simultaneously is the product of their individual probabilities. So the probability that an egg from the F1 generation, big P, little p, will receive that little p allele is 1/2, because it can only either receive the big P or it will receive the little p. Same thing from a sperm that has big P, little p, from that F1 generation. That it'll receive a little p is 1/2. It'll either receive the big P or the little p, which means the probability of a child being little p is a multiplication of the 2. So 1/2 times 1/2, which gives us 1/4. And if we look at a test cross of that over here, where we put a big P and a little p, a big P and a little p, and we were to draw it out. Oop. And we always want to do the uppercase letter first. And this is just due to making it a lot easier to read whether it's homozygous, heterozygous. What we see is right here, this little p is only 1 out of the 4. So that 1/4 probability shows. And this rule applies for dihybrid crosses as well. So here we have a dihybrid cross that is big Y, big R, little r, and we're crossing it with another big Y, big Y, little r, big R, little r. To go over how we can look at those gametes, the FOIL method is a great way to look at it. So big Y, big R, little r. How the gametes can occur is through looking at the FOIL method. We can look at it this way. You get the first two, which gives us one egg with big Y, big R. You do the outer, or first, outer, inner, last. So outer two, a big Y and a little r. Our inner two, a little y and a big R. And then our last two, which is our little y and our little r. Which means those are the possible egg and or sperm. Because if you look, both of the children that we have are heterozygous for the traits. So, a dihybrid cross is made when the phenotypes and genotypes of two independent alleles are analyzed, independent being the keyword. And the process is very similar to a monohybrid cross. So here, what they're doing is plant height and flower color. So the plant height being big T, little t, and the flower color being big W, little w. And so if we had a parent generation that were true breeding, so they would all be big T, big T, big W, breeding to a little t, little w, little w, we'd get a big T, big W, little w. And the reason for it is each parent can only give a big T, big W, because this one is big T, big T, big W, big W. It can only give that no matter how we go first, outer, inner, or last. Like, no matter what we do, it has to go that way. Same on this one for the big little t, little w. They can only give that little t and little w because that's all they have to give. Now, using the F1 genotypes of big T, big W, little w, and we're going to try to create what a child could be of this cross. So first thing we want to do is we want to do our FOIL. So we're going to go big T, big W, because that's our first. And then we're going to go into our outer, which is our big T and our little w. Inner, little t, big W. And then last, little t, little w. And because they are the same on both sides, it's going to be the same on this side too. So if we were to draw this out, we'd get big T, big big W, big W. Little w. And we would just do the cross like we normally would. So bring your T's down, bring your W's down, and across. And we'll draw this in class when you guys come back. So here we have another big T, a big T, and a big W, and a little w. And you just go through it, going a step at a time on all of them. If you look right here, we actually have that. We get 4 possible combinations of alleles, and we get this FOIL method, where it's big T, big W, big W, and then if you look over here, little t, little w in that bottom right-hand corner over here. And we get a ratio that is 9, 3, 3, 1, which means we have 9 who shows the dominant of both traits, 3 showing the dominant of one trait and the recessive of the other, and 3 showing the dominant of one trait and the recessive of the other, and then 1 showing the recessive of both traits. So this was with flower color, and for tallness. So we would have 9 that were tall and purple, 3 that were tall and white, 3 that were short and purple, and 1 that was purple and short. And this shows evidence of Mendel's law of independent assortment.